Hi guys, so we finished yesterday's lesson on this slide and I feel like I maybe didn't explain it very well. So I wanted to go back here to start today because we're going to go into meiosis more in depth today. So what I want to point out about this cell shown in this picture is that it has already had its DNA replicated, okay? And we know that because it has the sister chromatids, okay? Sister chromatids are present after the DNA has been copied, okay? So originally, the cell would have existed of one chromosome 1 for mom, one chromosome 1 for dad. So we'll call this one dad chromosome 1, mom chromosome 1, okay? Dad chromosome 2, mom chromosome 2, dad, I'm mixing my colors, dad chromosome 3, mom chromosome 3. Okay, so it had one from mom and one from dad and one from mom and one from dad. And then those got replicated, and that's when you get the sister there, right? And that's what we see here, chromosome 1, 2, and 3. That's what we see here in this diagram. Those sister chromatids attaching at the centromere. And then when they pair up, that pair from mom and that pair from dad, those are referred to as a homologous pair, one from each parent, okay? So sometimes this homologous pair idea and this idea from mom and dad are a little bit confusing because this is in our gametes, right? And so you think, well, my gametes are in my sperm. That's what's being passed down to the next generation. So how do we have mom and dad? Well, your gametes are made up of what your mom and dad gave you. So we kind of have like 3 generations that we're really working on here when we talk about meiosis, okay? So kind of keep that in mind as we move forth, okay? So let's go on into today's lesson. So when we take these gametes, so we have our sperm, right? And we have our egg and sperm, and we unite them in the process of fertilization, is the union of a sperm and an egg. It produces what is called a zygote, okay? Now these sperm and these eggs in humans would have 23 chromosomes, and so they are haploid. But when they unite, the zygote is going to have 46 chromosomes, and so it is considered to be diploid, okay? And that's just our one cell. A sperm plus an egg makes a zygote, which is one cell. The sperm and the egg are made by meiosis, okay? And you continue to make more by meiosis through, from puberty, beginning in puberty, and, through sexual, childbearing age, reproductive years. And then this zygote, right, once the sperm and the egg meets, it makes one cell. We all started out as this one zygote cell, and that cell will go on and divide by mitosis, because mitosis is responsible for growth, right, and development. And so this was our one cell that we all started out as, that came from our dad's sperm and our mom's egg, and it divides and divides through mitosis to become a baby, and then to become a child and a teenager, okay? And so that is the human life cycle. So today we are going to talk about meiosis. So remember, we said yesterday that meiosis goes through the cycle I P mat C, okay? And so we've got interphase, and then we have this 2 mat Cs. And so the first mat C is referred to as meiosis 1. Technically, that should be written as a Roman numeral, but I'm not going to really get crazy about that. And meiosis 2, okay? So we've got interphase first, and then we have our first round of cell division in meiosis 1, our second round of cell division in meiosis 2. So here we start with our diploid cell. It's got 2 chromosomes in it, one from mom and one from dad, okay? And so then those chromosomes replicate, and then we form the sister chromatids, okay? These are sisters, and these are sisters. If we're talking about these, those are non-sisters, as we discussed yesterday. Okay? So that's an interphase. Our DNA is replicated, so our chromosomes are replicated. In meiosis 1, that's the first round of cell division. So this one cell divides into two cells, okay? Each with half of the number of chromosomes. Well, not yet. That's going to be the original number of chromosomes, actually, okay? Half from what we were here in this previous step. And then these cells are going to each divide into two more cells. And down here is where they have half the number of chromosomes, or half of the amount of DNA from the parent cell. Okay? And so you see there's 4 cells formed here. And because they have half of the DNA, they are referred to as haploid. Okay? In meiosis 1, we had the separation of the homologous chromosomes. That's really important. Separation of the mom pair and the dad pair. In meiosis 2, we have separation of the sister chromatids, and that's important. Okay? And so that's kind of the overview of what's going to happen. All of these stages, as you're going to see in the next slide, maybe, are very similar to the stages that happen in mitosis. In metaphase, they line up on the metaphase plate. In anaphase, they pull apart. In telophase, you see the cleavage furrows start to form, the nucleus starts to reform, the spindle fibers, are broken down. And it looks very similar to the way that you remember mitosis to be. Okay? Let me take just a minute to point out a few differences. One of the major differences is in prophase 1. Okay? Prophase 1 is a major difference. In prophase 1, we have a process called crossing over that happens. Okay? And I'm going to talk about that more in just a second. So we're going to let that rest. But keep in mind, prophase 1, crossing over. If you want to draw that on that picture, draw that on that picture. Okay? But otherwise, you see everything that's the same. The nucleus starts to break down. The spindle fibers start to form. All of those things happen. Metaphase plate here. In anaphase 1, we have the homologous chromosomes separate. I'm drawing boxes around that both here and over here because that's important. Meiosis 1 is the separation of the homologous chromosomes. We're separating the moms, the red guys, from the dads, the blue guys. Okay? Notice how also in each of these we've labeled prophase 1, metaphase 1, anaphase 1, telophase 1. If you don't label it with 1 on your assignment today, it's wrong. Okay? Nobody knows what you're talking about unless you tell us. It's prophase 1, prophase 2, metaphase 1, metaphase 2. Okay? That's very important. All right? So after telophase 1 in cytokinesis, the cytoplasm divides. You form new cells. So these cells, both of them here, that were divided, we have the nucleus breakdown again. In prophase, the spindle fibers start to form. Metaphase, they're going to line up on the metaphase plate. Anaphase, I'm putting a box around this because it's important. In anaphase 2, we have those sister chromatids that separate. Okay? See them? The sisters are being pulled apart. And then in telophase 2, we have, in cytokinesis, we form haploid daughter cells with half of the amount of chromosomes. Okay? Again, that's really important. All right? So when we look at mitosis and meiosis, mitosis is very closely resembles meiosis 1. Okay? We have our chromosomes that are duplicated before interphase, and they're kind of paired up. Although you don't have a homologous pair in mitosis, homologous pair is unique to meiosis. They line up on the metaphase plate. You see that here. But you could be able to tell a cell that was in mitosis versus meiosis because of those homologous pairs. You don't see those homologous pairs there. Okay? And then they separate into two cells. Notice the cells here, though. Okay? they have the full amount of DNA. These also have the full amount of DNA. These are still paired up. Those are not paired up. Okay? But once we get to the end of meiosis 2, then if you look at the DNA there, it's considered to be half of the number of DNA, half of the amount of DNA, or half the number of chromosomes. Okay? Sometimes we will use the shorthand N to mean the haploid number. That's important. That's something that you could see on an AP exam. N is a haploid number. 2N then would be the diploid number. Okay? So for us, we would say 2N equals 46. N equals 23. That's for humans because our diploid number, we have 46 chromosomes. So 2N equals 46. N equals 23. Okay? That's important. This just again summarizes the differences between meiosis and mitosis. So both of them replicate DNA before in interphase, before it begins, before mitosis begins, and before meiosis begins. Number of divisions: mitosis has 1 division, meiosis has 2. We don't have synapsis of homologous chromosomes. That's the crossing over that I was talking about, and I'm going to talk about that more in just a minute. So no crossing over or synapsis in mitosis 1. We have that in meiosis, and as I said, that was in prophase 1. Okay. Number of daughter cells: we have 2 daughter cells that are formed versus 4 daughter cells that are formed in meiosis. Okay, because of that second division. And then this is for growth and repair, sometimes for asexual reproduction, as we discussed yesterday, versus meiosis is to produce gametes. Okay? So, when we talk about meiosis, there are 3 major things that allows for genetic variation. So you know that all organisms are unique, right? Or all humans, all of one type of organism, all of a species are unique. You know, I'm very different from my sister. Even though we both have the same parents and we, both got our DNA from our parents, we are very different. Personality-wise, looks-wise, all of those things. There's a lot of genetic variation even when 2 people have the same parents and the same gene pool. Okay? And so there's 3 major things that cause that. So the first is independent assortment of chromosomes. And so what that means is that during metaphase, when when chromosomes line up on the metaphase plate, okay, when these chromosomes line up here, they do so in a random way. So here you see it's not always all blue on one side and all red on the other side. This diagram is pretty simple compared to us because it only has, a diploid number of 6 versus our diploid number is 46. And so this looks pretty simple. But it lines up different on the metaphase plate every time. And so those chromosomes, the way that they are sorted or shuffled out to the cells differs every single time. Okay? If you took the same zygote, the same fertilized egg, which you can't really do this, but if you did, and you took the same fertilized egg and you took it through meiosis, you could have different combinations that are produced because they line up different on the metaphase plate every time. Okay? And so that's kind of interesting there. So that is independent assortment. The way that the chromosomes line up on the metaphase plate is different every time. And so those the traits, the alleles, assort independently. Okay? So this diagram is kind of supposed to help explain it. This is a simple diagram again, but you see that we have different combinations based on the way that the chromosomes lined up. Here we've got a red and a blue. Here we've got 2 reds. So down here you've got a combination of 2 blues or 2 reds. Down here you have different combinations. So again, this is a very simplistic diagram, but you see that those chromosomes assort independently, allowing for different combinations in the gametes. Okay, so these gametes, combination 3 is going to be different from combination 4. Okay, and so that's just one. I said there's 3 main things. This is just one of those things. Okay, independent assortment. The next one that I've been holding out on is crossing over. Okay, so what crossing over is, and it shows it in that other picture, I'll go back to it in just a second. Non-sister chromatids come together in something that's called synapsis. Okay, let me show you back on this other image right here. These two homologous chromosomes pair together, and they come together like this, and that's called synapsis. This shape that they form, where they kind of form like an X that you see here on the ends, is called a chiasmata. That's singular. Chiasma would be the plural form if you see it written like that. But they synapse or come together, and they form a chiasma, and they do crossing over. And so what crossing over is it's kind of the exchange of parts of the sister chromatid. So you see this, they exchange these pieces of their legs. Okay, I'm calling them legs. They're not legs, they're sister chromatids. And so you see that here. So this blue chromosome, this daddy chromosome, is not all daddy anymore. Some of the genetic material is from mom is now embedded in there. And the same is true over there. This diagram shows synapsis in 2 places. Sometimes synapsis will happen in more places than that. It can happen in a few places on a chromosome. I think between 1 and 3 is what I remember, if that's correct. And so what we see here is once these chromosomes separate, these daughter cells are all different. Okay? These daughter cells are all different. Some of them are recombinants. Not all of them are recombinants. Those are not recombinants. But these 2 are recombinants. Okay? And this picture here in independent assortment doesn't show those recombinant options. Okay? So crossing over is another major source of genetic variation in gametes. Okay. The last source, number 3, if you're keeping count, of genetic, well, I guess there's 4, of genetic variation is random fertilization. And so what that means, that we know that every sperm is different and every egg is different, and fertilization happens randomly. Whichever, sperm gets the egg first, that is random. And so when you take all of these combinations together, scientists, and I could go through and tell you all the numbers, and it's like 2 to the 23rd times 2 to the 23rd or something like that, is 70 trillion different combinations in a zygote. Okay, after crossing over, after independent assortment, after random fertilization, there's 70 trillion possible diploid combinations of a zygote. Okay, which makes us all very unique. Now, on top of that 70 trillion, though, we still have this guy that we haven't accounted for, and that's mutations. So mutations can happen in the gametes, but we also know that mutations can happen in the DNA as the gamete continues to divide and reproduce. Okay? And so a mutation is any change in the DNA, and that's how we get genetic variation. Mutation is considered to be the original source of genetic variation. Okay? Because if there's not any mutation, then every single cell is the same. And so mutation is considered to be the original source of genetic variation. So once you add mutations on top of this 70 trillion, there really is nobody else like you.